a PhD student here at the Max Planck Institute for Chemical Ecology in Jena. And I'm here to talk to you today about whether or not toxic animals are being honest with their colors. And you can see these lovely little bugs here in this picture, which I will be introducing you to shortly. But first, let's talk about how some animals advertise their defenses. So when we say defended, we mean a chemical defense. This can mean poisons, venoms, or just toxins in general. And aposematism is when this defense is connected to some kind of signal that warns predators that this defense exists. So traditionally, these signals were considered to be colors. So for example, you can see the black and yellow wasp, or the black and white skunk, or the yellow and blue-ringed octopus. All of these have very conspicuous colors that warn you that they are toxic or very unpleasant to eat. But they can also be strong smells or sounds. For example, the rattlesnake is not particularly visually striking, but they make a very, very loud noise when you approach them, which warns you that they are not going to be very pleasant to try and eat. And this is kind of like a stoplight for predators. So it says, please don't eat me, you're not going to like it. And it works quite nicely as an analogy because most of the classic aposematic colors are considered to be red, yellow, black, and white. So lots of very smart people have done a lot of maths, and they've found out that predators would learn to avoid toxic prey much easier if they were all exactly the same, which kind of makes sense intuitively, right? So if you saw something that was red and circular, for example, and you had a terrible time trying to eat it, if you came across anything that looked like that, you would then not want to eat it as well. They also remember this association for longer. So it's kind of a mystery then that in real life, in the wild, there's lots of variation in both color and toxicity in aposematic animals. And this is a nice figure from Andrew Davis's paper on monarch butterflies. And he found that the migrant butterflies had a deeper and darker red coloration in their wings. So why would this exist, this variation? Well, luckily, somebody did a review paper on it 2 years ago, and they've looked at lots of different reasons why this might be the case. But first, let's talk about what kind of variation does exist. So there's what we call morphs, which is when something looks very different. For example, within one ladybird species, there might be black and red ladybirds with different numbers of spots. There can also be continuous variation. This one in the bottom right, which I've been looking at, and that's when there's sort of a gradient scale, so shades of red or brightness in coloration within the same population. So why does this variation exist? Well, there's plenty of different reasons why this could be the case. It could be that there's different predators which exert different predation pressures on the population. It could be that there's physiological reasons. For example, melanin, a very common black pigment, has effects on thermoregulation. It could be disease. So for example, more ill animals, maybe they're not able to generate the colors, or maybe they're not able to sequester the toxins. It could be sexual selection. So in many species, females prefer brighter males or more shiny or whatever it is, and this could definitely affect their coloration. And it could be resource availability. So that's, do these animals have the resources available to make or sequester their toxins and to generate their coloration? And this could lead to something called honest signaling, which we can look at now. So honest signaling, there's two different ways of looking at it. There's qualitative honest signaling, which just means I am red and I'm toxic, so don't eat me. It's this signal that's accurately saying I have a defense. Something that would be not honestly signaling would be something like a hoverfly, which has got the coloration but not got the defense behind it. So by definition then, all aposematic animals are qualitatively honest signaling because they all have a signal that is signaling a defense. However, what's more controversial is this idea of quantitative honest signaling. So that's the idea that there's a correlation between the coloration and the toxicity. So for example, within my population, I'm more red than my co-species, and therefore I'm more toxic, so you should definitely not eat me. And people have looked for these correlations in the wild in quite a few observational studies. And it has been found, for example, across species of sea slugs, that the brighter species were the most toxic. It's also been found across different populations of the strawberry poison dart frog. And it's been found in paper wasps that the brighter wasps had larger venom glands. It's been found in ladybug eggs, although another paper found that it didn't really exist in the ladybird adults. On the other hand, there's actually been a negative correlation found across different species of poison dart frogs, with the most toxic species being the least conspicuous. And recently there was a paper on the six-spot burnet moth, which found no correlations whatsoever. So there's some evidence for, some against, and some neutral for there being quantitative honor signaling in aposematic species. So one explanation for why these correlations might exist is a model that was developed by one of my supervisors, Professor John Blount, at the University of Exeter in the UK. So his idea is that many pigment molecules are also known to be antioxidants. Antioxidants are molecules that help combat oxidative stress. When sequestering toxins or creating them de novo, perhaps animals then face increased oxidative stress, and they need these antioxidants for that. But then there would be a trade-off because they would also need these pigments for their signaling, for their coloration. So according to the different resource state of the individual, this could lead either to a positive or a negative correlation between the signal and defense, so between their color and toxicity. And this could explain quite a lot of these relationships that we see in this slide here. So in my PhD, I'm looking at whether or not this model could be true for the large milkweed bug, Onchopotus fasciatus. Does the large milkweed bug show quantitative honor signaling? So first, let me introduce you to the milkweed bug. Unsurprisingly, it feeds on milkweed, which is a genus of plants in the Americas. Milkweed plants are toxic themselves. They create these toxins that are called cardenolides, which is a type of heart toxin. So if you eat too many of them, your heart will stop. These bugs take the toxins from the plant and put them in their own bodies, something we call sequestration. And these bugs are very bright red and black. You can see that on the bottom, that's an adult. They also aggregate as nymphs, so when they're small, they cluster in huge amounts around these seed pods, which is what they feed on. And most importantly for me, the populations vary in toxicity and color in the wild. So they seem like a very good species to test this model on. So unlike most of the other studies, mine has been experimental. So I have raised these bugs on sunflower seeds, which they can be raised on, and I've added my own toxin, so I can control how toxic the bugs have grown up to be. I looked at correlations between toxicity, oxidative stress, and coloration. And if the model was to be the underlying mechanism in this species, I would expect that more toxic bugs would face more oxidative stress, and that a correlation would exist between color and toxicity, so between their redness and their sequestration. So what did I find? Do bugs face higher stress when sequestering? The answer is no, not at all. I looked at several different aspects of oxidative stress, and none of them showed a definitive increase in stress from having more toxins. The only really clear trend is the one I'm showing in this graph here, which is that when they get older, they get more lipid damage, which is what the MDA stands for, malondialdehyde. So L4 and L5 are the nymph stages, so before they molt into adulthood, and A1 and A3 are both adults. But this isn't overly surprising. Almost every animal faces more oxidative stress as they get older, but you can see that there's no treatment differences here. And the big question, are bugs more red as they get more toxic? And it is also no. So I did not find any correlation between color and toxicity. I also looked at brightness and toxicity and found no correlation there either. However, there are some pretty interesting life stage differences. So you can see in the legend there, L4 and L5 are those little crosses and squares. Those are higher up on the graph, which means that they have redder coloration. This also tracked with the luminance, they also were brighter. You can also see that the adults, the ones at the bottom, the triangles and circles, are on average more to the right-hand side. And that means that they were more toxic proportionally than nymphs. So what do I draw from all of this? Although my little friends, the milkweed bugs, vary in toxicity and color in the wild, they do not seem to be showing quantitative honor signaling because the expectations of this model were not fulfilled by my experiments. However, younger bugs tend to be brighter and redder and less toxic than adults. And this is really interesting. These life stage differences could be due to behavioral differences, for example, because the younger bugs aggregate. Perhaps they don't need to be as toxic because they're all in a group. It could be the type of defense. So one of my coworkers is looking more into this, but adults and nymphs actually secrete slightly different types of cardenolides. And also the mode of defense. So they release their secretion, their toxic secretion, from different points in their bodies. perhaps they're then aimed towards different predators. So I'll be looking at all of this further on in my PhD, but for the moment, thank you all very much for listening. Thank you to my collaborators in Hohenheim, my supervisors, and my group here in Jena.
Question.
Mm-hmm.
Do you think that there's no correlation is only for the milkweed bug, or can this be generalized?
It's just for the milkweed bug. So if you look at, there's lots of evidence, right, for and against these correlations existing in nature. And just because this model is, perhaps it is the underlying reason for some of these correlations, just because I showed that it isn't in my species doesn't mean that it isn't true for other species. So that's what's cool about my research, is that it adds to a very growing area of science, because the science is really not at all settled on this point. So I think this model could definitely be true for other of these species, but perhaps the milkweed bug isn't using pigments like this. Perhaps it is not as antioxidant as another species.
Say bye.
Okay, thank you everybody. Bye-bye.
Nice.
That was real fast. Oh well.